So once again, good afternoon, viewers at home. My name is Advanced Michael. I'm the team leader of Om3ga Bio-edutainment, where we are basically concerned with education, entertainment, enrichment, entrepreneurship, career and mentorship, romance and relationship, sport and culture, nutrition and health, and we also advocate for community people. That's not the business for today. This is going to be a continuation in our educational aspect of this Om3ga Bio-edutainment platform. And today we are going to continue from where we stopped the other time on our course, which is called Toxicology. Why I'm staying behind the camera is just that what you have on the left hand side on the marker board, that is from 1 to 4, that's going to serve as a kind of review. That is where we are coming from. That aspect supposed to have been taken before, and which I think part of the earlier video must have addressed, if not all, some parts of that part. And the section B is going to come a long way, and that's going to be the new ground that we are going to break today. Again, don't forget that you should hit the notification button, like this video, make comments wherever you have critical feedback from the comment section. Don't forget that we are starting, just like I said, from the left hand side, which has to do with the overview of toxicology 2. If you will not forget, you will remember that toxicology 1 is a principle and what we can call the introductory toxicology. That is telling you that at this point in time, we should not actually be introducing you again to some basic terms and terminologies in this course of toxicology 2. That this is going to be a kind of an advancement over the foundational toxicology, and it is going to be a focus in this toxicology 2. We are going to focus mostly on the mechanism of toxic substances. How does it actually interact with our biological system? And don't forget that our biological system could be prokaryotic, it could be eukaryotic, and it could also could be man, can come from even the domestic animal around us. So that is the reason why we are going to be concerned with what is going to be the substance toxic to us, and what is going to be composition. How is it going to, what are the mechanisms of its action, and how can it actually be controlled, giving a kind of risk assessment. So just like I said, this is going to be a deeper understanding of how toxicology is actually being studied at that level, and that is what is going to help us as a kind of overview of what toxicology 2 is actually going to be. So that means this is going to be a more advanced, it's going to be more modern, it's going to be a toxicology that is made for nowadays. And we will also find an attempt to be able to incorporate this toxicology as a course, to integrate it into the other aspects of a biological system. In fact, there will be a time that we just have to look for a way of studying it in terms of epidemiology, looking at it from an environmental toxicological perspective, looking at it in terms of how that risk or how that substance is going to be able to communicate. So that means we are going to introduce some elements of biological courses, biochemistry, statistics, computer programming, and some other parts of other disciplines as well. So that is what I just want to say about how this advanced toxicology is and should be overviewed. That is, it is going to be more advanced than what we have actually learned in introductory aspect of toxicology. But don't forget that I said toxicology is actually the study of toxic substances, harmful substances, risky substances. And I told us that even at this level, 93% of what form a kind of toxic substance is actually domesticated in our homes. So man cannot actually run away from exposing himself to toxicants or toxic agents. So then I told you that there is no substance that is actually safe to be regarded as non-toxic, depending on the dose or the dosage, and depending on the time of exposure and the type of the substance that is actually being involved. So it is the science of poison. It is the science of studying what are poisonous substances. And just like I told you that poisonous substances could be of different sources. It could be from biological agents, it could be chemicals, it could be environmental, it could be from anywhere. So that is that about the introductory part of this course, which has to do with a kind of overview, general overview of this course. We need to consider all this. Now, in the earlier video, we have also considered what are the advanced toxicological mechanisms. And at that point in time, we consider that what are the pathways by which toxic substances can actually get into our system or can actually get to be studied, and how can we actually classify them based on their various mechanisms. The first one we actually consider was cellular mechanism. Substances that will interact or that we have is basics interacting with a kind of cell membrane, which thereby alter signal transduction or disturbing other poisonous compound or poisonous cellular processes such as protein synthesis, cell division, or apoptosis. I told you that apoptosis is a process whereby cell will commit a kind of suicide, that is self-death. So there are sometimes that some of these substances can be so harmful to the fact that they will not directly harm or lead to what is going to actually affect organism, but is going to interact with protein synthesis. So any toxicant or any substance that can interfere with cell membrane, that can have its effect on altering signal transduction. I told us that signal transduction is a way whereby cells do communicate to each other, that is cross-talking. So that can interfere with that can interfere with pathways or other cellular processes such as protein synthesis, transcription, translation, cell division, or the way or the mechanism by which microorganism or by which our body carry out their cellular vision. So any interaction that involves that can be classified as an advanced toxicological mechanism. Then the second one is we also have molecular level disruption. That is a time whereby you have toxic agents or toxic substances that has to interfere with the biomolecules like DNA, that is going to interact directly with proteins, that's going to interact with lipids, and that's going to interact with other enzymes. So thereby, this kind of molecular interaction will lead to a kind of mutagenesis, that is going to initiate mutation, and the elements, I mean, in the elementary stage of cancer is called mutation. So whereby it can also initiate carcinogenesis or other pathological conditions. So this is nothing but it is regarded as molecular mechanism, that is molecular level disruption of that. Then you can also have the third category, which is a receptor binding and enzyme inhibition mechanism. This is when a toxicant or some of these toxicants will exert their effect or their impact by binding to specific receptors. Specific receptors. Don't forget that immediately after every metabolic pathway or every cellular processes or activities, the cell will begin to regulate. That means, and it can regulate that at process level. It can regulate that at step by step level. So, and what that one can do is that it could be inducible, it could be repressible, or it could also be a way of activating it. So, whenever you now have some of these toxicant agents binding directly or inhibiting directly key enzymes of metabolic pathways, physiological function. For example, it is known that organophosphates, they exert their poisonous effects by inhibiting the acetylcholine esterase, or which is found, this is an enzyme that is found in the nerve cells. That is going to have a direct effect on the central nervous system. Example of this is most of the lead substances that is known to induce a kind of brain damage in children and some other, and even in adults as well. Then the fourth one is oxidative stress, and those ones that are involved in free radical production. This is a major mechanism of toxicity, and this one is what leads to generation of what is known as ROS, that is reactive oxygen species, that is going to cause oxidative damage, which is going to have its effect, eventually, it's going to have its effect on lipid, protein, and DNA. The example of a receptor binding, before I forget, is that of some of the agents, carcinogenic agents that will go and intercalate, they will go and interact directly with the DNA, thereby causing mutation, thereby causing damage, genetic damage in form of a site mutation or site mutagenesis to the DNA. And at the end of the day, it is going to eventually lead to manifest in the next generation, which is going to be nothing but is going to be genetic manifestation. So that is the first aspect that has to do with the toxicological mechanism. So I told you that they could be of four types. Cellular, they could interact at the toxicants can interact at cellular level. It can be in form of a molecular binding interaction. It can be a kind of receptor binding and enzyme inhibition procedures. And I've given you different examples of that. And the last one, which is oxidative stress and radical production. Now, just like I told you in the introductory part of this course when I was talking about number one, which is overview of toxicology. There is nothing that we can do at any point in time in this course if we will not actually relate it eventually into the effects on public health. Just like you hear from the name public health, that is is going to have effects that could be epidemics, it could lead to pandemics, and it could also lead to every other outbreak that we can actually lead to, depending on whether the agent of the poisonous agent is biological, whether it is chemical, or whether it is environmental as well. So that is going to lead us to um, toxicology in public health. Now, if we say our environment is safe, what do we actually mean? That means the public health or the environment is free from contaminants. The environment is free from pollutants. So, be it whatever it is going to be, whether it is going to be biological, source is going to be biological, whether the source is going to be physiological, whichever source is going to be, that it must have its effects on the environmental. And in some occupations, we see that some occupations are more hazardous than others. So, for example, people that actually work in the production of car batteries, now you will know that occupationally they are more exposed than people that work in offices where paper is being produced. Those that work in factory where plastic is being produced will be quite different from, they will be exposed to different chemicals. They will be exposed to different toxicants more than somebody who is just sitting down in the office, who is only a lecturer, or who is only just in the in academic environment. So, and that is the reason why that whatever risk assessment we are going to carry out, whatever effort we are going to do in toxicology, if we are not considering the public health importance, that means we have not been able to do anything at all. And don't forget that how related is epidemiology in toxicology. So this is a method of finding how diseases could be treated, diseases conditions could be treated as a result of their health outcome. That is, whatever happens, you are still going to trace it back to what could be the health impacts of these toxicants at the end of the day. So, whenever this epidemiology as a course will actually assist us, don't forget that I told us that toxicology cannot be studied in isolation. You have to study it with other parts, with other disciplines. So, and that is where we are now talking about epidemiology now. So, you need to look at that toxic, you need to identify the toxic substance. Having identified it, you also need to know what could be the overall health impact of that toxic substance. So you are still going to lead you are still going to link it again to the propensity of that toxic substance to be able to initiate some health condition or developmental disorders or congenital diseases. Okay, so that is why epidemiology is very, very important in toxicology. Very, very important. Now, how related is also environmental toxicology? So, how related is that? So, this is very related because this is the field that is going to look at how infected, let me use that word, how infected or how safe or how harmful that our water could be. Our water including the soil, including water, including this, that is our environment safe? Well, what are they safe from? Are they safe from pesticides? Are they safe from heavy metals? Are they safe from industrial chemicals? Are they safe from other kind of pollution or pollutants? So that is why environmental toxicology is very, very important before we'll be able to do well in toxicology, because we have to lead it back. We have to link it together to its effect, its overall effect on public health. Then again, this third aspect, which has to do with risk communication. So it is one thing to assess the risk. It is another thing to be able to report back to the community. You should be able to publish your data. You should be able to inform government policy makers. It is also very, very important for you to be able to communicate your findings to the external world or to the people of the community. Just of recent, some of my project students were actually interested. People do take a lot of energy drinks. So I now ask them to test how risky or how safe could it be in albino rats. And at the end of the day, they came up with their findings on the vital organs, which include kidney and all this one. And that was when they were able to tell us how safe it could be and which one is actually more safe than other. Is it going to be? I can still remember that they made use of different types. They collected different types of energy drinks, and they were able to assess in terms of varying the concentration. And at the end of it, so having gotten that, that another thing is, how can they communicate the risk to the community people? But don't forget that once you are communicating this, you also need to be very, very careful because of the ethical issues or ethical implications, so that somebody will not come and get you arrested, that one way or the other you are damaging their market or the market value of their product. So that is the aspect that has to do with risk communication. Then again, the other aspect that is also very, very important in public health, toxicology in public health, is the aspect of vulnerable population. You need to know that before you will report any substance to be toxic or not, that means you have to recognize some of the vulnerable populations. And the vulnerable populations are the older ones, that is the old men and women, the children, and as well as pregnant mothers. So you need to know what could be the effect of all this. So the elderly, you need to know how susceptible are some major group of people in that population. How susceptible or how vulnerable are they, especially children, the elderly, pregnant women, and as well as what are the other underlying conditions or pre-existing condition that can make these vulnerable people to be a kind of focus in public health. So the fourth aspect of the review of the aspect we considered before is regulatory aspect of toxicology. So you should know that there's nobody that is going to be too safe where there is no regulation that will guide the reportage, that will guide the conduct of conducting assessment, toxic testing, and risk assessment in any community. So that is why we will consider what are the regulatory aspects of this course, which is advanced toxicology. The first aspect is toxicity testing. So this is going to involve you determining how safe a substance is, how safe it is in terms of is it going to be safe in acute, is it going to have acute effects, is it going to be subchronic, or is it going to be chronic? So, and that is why, just like the experiment or the research I said my student just carried out, is a kind of a toxicity testing assessment to look at vary it in terms of concentration. So those ones that we impact, we have some acute impact, they will know. And those ones with chronic impact and acute impact, they will know. And that is why in vitro studies or in vitro assay is very, very important. And nowadays, informatics, bioinformatics has actually helped us to be able to use some computational analysis in order to identify. So instead of us wasting money, go to the lab, real animals, real this and real that, another thing we can do, another testing that we can do, or another experiment that we can do, is to conduct in vitro studies, is to conduct computational study that will give us the how safe it could be. So, and that's why at the end of the day, even when we are considering drug testing, we also need to consider do a kind of toxicity testing. So, and there are some various databases that are now devoted to test the effects, the toxicity effects of certain chemicals, not only chemicals, biological agents in form of drugs, vaccine, and what have you. So that is under toxicity testing. So that is a part of what they will ask you. They will ask you the regulatory this that you have to report it. Then the second one, which has to do with risk assessment. I told you this is a structured process that's going to evaluate the likelihood of the adverse effects. So you want to know the possibility, whether is it going to be safe or it's not going to be safe. If it's going to be safe, in what quantity is it going to be safe, or we should not just consume it at all. So don't forget that in some part of the world, they have banned the use of chloroquine, aspirin, and but up to today, in some part of the world, some are still using it. So, and this is because maybe the risk assessment studies has not actually implicated them to be causing any effects in terms of that. So then the fourth aspect, which has to do with your relationship with regulatory agencies. So regulatory agencies like environmental protection agencies, you need to know. So before you will be able to report how safe is a toxic substance, you should be able to know what some of these regulatory agencies are going to be saying after that. For example, each occupation has its dosage, safe dosage. They have some safe dosage which has to be this. So all these regulatory agencies are very, very important in toxicological studies. So which you have to know. Then the last part of this, which has to do with the Toxic Substances Control Act. There are some laws that have been promulgated and which some countries in the world are part of those that actually signify they are signatory to that kind of law, and that is why regulatory agencies, you have to know what they are saying about all these toxic substances. So just like I said, behind this the camera, I've been able to actually tell you and give you an overview of what we have done in the past, and that is what we have just done now. That we just try and review what has been done in the earlier lectures, and this section B is what we are going to do now in the next moment. So the second aspect of it, don't forget that I told you that you cannot regard any substance, no substance should be regarded as a poison or as toxic if you are not considering the dose, if you are not considering the dosage, the route of exposure. All these you're supposed to have come across them in the earlier video. So, and that is what we are going to do today. We want to look at a dose response relationship. Dose response relationship. Okay, and the principles. What are the principles of dose response relationship? So we are going to talk about dose. We look at what could be the response. What are the key principles of a dose response relationship? What do we mean by threshold dose? What do we mean by MTD, that is maximum tolerated dose? Then what is a potency? So after potency, and what do we mean by efficacy? If you are saying any substance, before any substance can be said to be effective, so what are we saying about it? So then B is going to be different types of dose response curve. That is, if you see, this is graphical representation of how safe some certain dosage could actually be. We have some that will be in form of a linear dose response, and some will be non-linear dose response, and some will come in form of sigmoid, that is S-shape, and there are some that will also come in form of homeostasis. Okay, that will come in form of homeostasis. Now, what do we actually mean if we are talking about dose? So dose refer to the quantity. Dose refer to the amount to be taken to determine whether that substance is safe to be taken or it is not. So that is a dose. It refers to the substance. It refers to the quantity. This is actually talking about the quantity. The quantity of toxic substance of which an organism is exposed to. That is the amount. So for example, before you will say whether lead, or let me say heavy metal like lead, so you have to consider the dose, that is the amount that individual is being exposed to. So for example, every day of our life, man begin to get himself exposed to different types of chemicals, but in different dosage. So not until it has exceeded that dosage before it will now be regarded as being toxic. Then what do we mean by response? Response is just that what is going to be the biological effect. So the biological effect, that is, what is the effect of this lead on our biological system? Is it to our skin? Is it damage to our skin? Is it going to be damage to our respiratory organs? Is it going to be damage to our kidney? So that is what is actually called the response. That is response, which refers to the biological effects that will determine the severity, of any dosage or any quantity of lead that's going to be effective. That's going to have effect on that. So, and some of the key principles of those relation reaction. So you might come across threshold dose. Threshold dose. This is tolerable. This is going to refer to the tolerable dose, that is not going to have any biological effects. So that is what is called threshold dose. It's not going to have any observable. It doesn't have any observable effects. So for example, now if you are considering lead in terms of how the body or how the human system is going to react to it, that means we are going to consider the amount, the threshold dosage, that the amount that's not going to have any effects, be it acute effects, be it chronic effects, or be it subchronic effects. So that is the threshold dosage. And the next one, which is maximum tolerated dose. So maximum tolerated dose, that is just from the word maximum, that is highest. That's the highest dosage that's going to have effects. That is the level. So for example, if the threshold is 20 milligram, if the threshold effect is 20 milligram, and if they now say, okay, the NTD is 150 milligram, so that means this is the highest that your body can actually tolerate. So, and it's going to have effects. So that means once the concentration has reached this, it's going to have serious effects on that organism. So don't forget, after maximum dosage, the next thing that we also need to know is potency. We need to consider potency. Potency is the amount to be consumed, or the amount to be taken, that is going to be very, very effective, potent. That's going to be very, very effective. So if they say something is potent, that is, it is effective. So that is that. Then efficacy. So efficacy is just the measure, of the maximum response. Measure of the maximum response that is achieved irrespective of the dosage. Irrespective of the dosage. That is efficacy. Okay. Now the other part we are supposed to take is what are the types, different types of those response. So today, what I will just do is just that I'm just going to list them. So whenever we now come back for the next topic, that's when I will now give us some of the graphical representation of this one. But don't forget that we said we have one linear. Linear is when you have linear is when you have this is dose, and this is a response. Okay. So if at the end of the day you now collect your samples, and you now conduct what is called a risk assessment. So if you now have something like this, that is like this. Let's say you have something like this. If you have something like this, so by the time you now want to plot this graph, if you now have something, if you now have it like this, if you now have it like this is what is called linear, that is straight line, or it can be like this, linear dose response. That is linear dose response. Non-linear is when you have something like this. This is a dose, vertical axis, and this is a response on the horizontal. So by the time you now plot what's going to be your risk assessment values, and if it is now going to, if you now have something like this, so that by the time you start plotting it, you now have something like this. This is called what? Non-linear dose response. This is A, this is non-linear. So this is A, this is A, this is B. C now, which is sigmoid. Sigmoid is when you now have the curve like this. Sorry. So you know it's going to form something like this. Going to form something like this. So this is what is called a sigmoid. That is a sigmoid. That is C. D now, which is called hormetic. That is the one that is having J shape. J. So by the time you now plot it, you now have something like this. So this is a G shape, and there are different. Now let's now look at which of these can give us a linear dose, and which one can give us non-linear, and which one can give us a sigmoid as well as this. So when you at least linear, that means there is no threshold. Any risk assessment that doesn't have threshold dose, or any substance that has no threshold, that is going to be linear. That's going to be linear. So if you are considering how safe a chemical is, and that chemical doesn't have any tolerable dose, it is going to form linear dose response. And that of non-linear, so that means it is going to be, that is B. That is non-linear. That means it is going to fall below. It's going to fall. Most of your diagram will fall below the threshold. It's going to fall below the threshold dose. So anyone that fall below threshold dose will give you non-linear dose, and anyone that has a gradual increase, this is a gradual increase, so it's going to form sigmoid. Gradual increase, that is, it's just going to pick up gradually, so eventually it is going to form S. Don't forget what I've said now. The linear, that means there is no threshold dose, no TD. No, so that one is going to be linear. And B, that means it is the graph or the risk assessment study is going to fall below the threshold dosage, and that of the C, which is a sigmoid, is going to be a kind of gradual before. So the next one is hormetic, which is a J shape. So in some cases, low doses of substances, they will have beneficial effect or stimulatory effect. So those ones that the threshold at the end of the day is going to be stimulatory or is going to be beneficial, is going to form a J shape plot diagram, so to say. So just like I said, we are going to stop here today, and in the next video we are going to continue our discussion on the toxicology 2, that is advanced toxicology. Don't forget what we have done so far today, that the business of today has to do with studying the dose response relationship, and we look at what a dose is, what could be the response, and what are the basic principles of a dose response relationship. And we look at threshold dosage. This is the tolerable amount, and we look at what is MTD, that is maximum tolerated dosage. We also look at what is a potency, what do we mean by efficiency, and we eventually look at what types of those relationship curves that we can talk about. We look at linear, that is any study, toxicological study, that the substance you are considering has no threshold dosage, so that's going to be linear, straight line. And the second one that has to do with non-linear, that is telling you that any toxicological assessment studies that falls below the threshold dose is going to give you linear. And sigmoid curve, that is when there is a gradual pickup or gradual increase in the curve. And I told you about homeostasis, that is when the substances you are now considering is now, instead of it to have harmful effect, it is now having a stimulatory effect or activating effect on the system that you are actually considering it. So what is going to give you the shape that is going to give you at the end of the day is going to be J shape. Please keep in touch, like and share.